Did you know that the Amazon rainforest is never, ever quiet? Even if you turned off your headlamp in the middle of the night, tried to be as quiet as you could be, you'll still hear sounds. Even in the dead heat of the afternoon, when you're expecting everything to be taking a siesta, and loads of primates do, there are still things calling in the rainforest. And that's because the Amazon is just staggeringly biodiverse, right? It supports an enormous amount of life and an enormous variety of it. I have the great honor today of representing a team of scientists that do work in this region. And I am really glad to be able to share their most recent achievements as they sort of solve problems for this region, one pod 5 at a time. So most of you have probably, in any context that you've heard about biodiversity, always had the follow-up, right? Which is that there are things out there that we do that severely threaten biodiversity globally. So pretty much every time you hear about how amazingly biodiverse a place is, you're hearing about how it's endangered, and how we're pretty much losing things faster than we can even record that they exist. So when I see a slide like this, an infographic like this, and I see numbers like 94% of populations are declining or being lost in a particular region, I make one really common assumption, and I assume that we, the scientific community, are actually aware of what 100% is. We know what's out there, right? But do we really? And if we're wrong, exactly how wrong are we? A group of scientists in 2020 decided to sort of explore this a bit. And this is just one example. There's many others out there. They looked at the eastern Guyana Shield, and at frogs in particular. So they went out and actually did a whole bunch of sampling in areas that they thought were undersampled. They took samples, DNA barcoded them, assembled sequences, but they also scoured the internet and all databases out there for any Anuran sequences from that region, and they tried to estimate, what they thought would be an unbiased way of figuring out whether how many species there were out there, right? So they ignored what people declared sequences to be coming from. They actually clustered all the sequences together and tried to create trees to figure out how many OTUs they could find. And classically, it was thought that in that region there was about 500 frog species, but it turns out that they were very, very wrong. And how wrong? There were over 2,000 species, so about 4 to 5 times off, right? So in Peru, my colleagues at the at the National Museum of San Marcos in Lima, went ahead and tried to do this kind of analysis for the country. And these are people who are responsible for maintaining the lists of birds for the nation of Peru, right? So the bird department looked on the internet for any sequences out there for any of the species of birds that were on their list, and they found that about 21% of the bird species had absolutely no genetic material sequenced and available at all. Then when the mammals team went ahead and did this, they found that that number was about 45% of any mammals. And what's most interesting is that even when there were sequences out there, these were not necessarily of the species from Peru, right? So it could be the same species, but sequenced in Brazil or another country. And so when you think about what was happening in the Guyana Shield with frogs, you can imagine that we're probably really not very accurate in terms of how much avian or mammalian biodiversity there is in one country, because we haven't been sequencing locally. So the reason, I think, that there are these differences, these inequalities, these gaps in our knowledge, is because science mirrors society, and society is a product of history, right? So we can see, and there's so many different examples of this, but I'm just pulling out 3 things. If you look at the study on the left here, where you can see a plot of authorship and GDP, of about 10,000 papers, it's just much more likely that somebody would be an author if they were from a country with a higher GDP. If you look at Nobel Prize winners by continent, you can see that the vast majority of them are in the global north, and having just 1% from South America or 3% from Africa is not an accident, right? So one group of people colonized another group of people, and this is just a reflection of that history. Now, even in the global north, when you look at other types of distributions of why these prizes are given out or to whom they're given out, you can see that there are these other ways in which science has been sequestered. So who gets to do this stuff is not an evenly distributed thing, right? So women there's such an enormous gender gap here that, I mean, look around you. Look at the number of people, number of qualified women you know, excellent scientists you know. It's almost like they have to put some special effort into trying to maintain this kind of gap. So when you take this down and look at it in terms of conservation genomics, it's actually very hard to try to figure out where the gaps exist. So don't hold us to these exact numbers. We tried to do our best here. We looked at the NCBI genome sort of collection or database out there and tried to establish the country that did the sequencing and then also the country from which where the animal actually normally lives, right, or the animal or plant. And you can see that there's this massive difference. About 91% of genomes sequenced in the northern hemisphere, even though 43% of all of those animals are coming from the global south. So in the context of doing this kind of work, I've had to face the fact that I was very much a part of this unequal paradigm. I've done lots of exporting of samples myself, and I still have to fight it all the time. I have to try to make sure that I can do science locally, and it isn't an easy thing to do. But when I began to do this work, I started to get a lot of questions, right? So what I was proposing was, why take samples to labs if you can take labs to samples? Let's flip the paradigm, do it differently. And I would get some pushback. So here's a few things that I heard. One is that conservation genomics has to have an established, expensive urban laboratory. So you actually have to move samples out because these laboratories don't exist in most of the countries where these specimens are actually collected or where the animals or plants exist. The second is when you propose to actually set up a lab, that is different, right? It's not an urban laboratory. It's something that's in a field station, for example. You're always told that these labs will just never really be good enough, right? They're never going to be able to take you all the way from sample to sequence directly on site. You're going to need outside help at some point. And this is where Nanopore has completely changed the game. So one of the biggest obstacles has always been, whether somebody can eventually take DNA or RNA and get it sequenced locally. And now you actually can. It's within reach. It's very doable. And we've been doing it, and I hope I can show you how. But the most pernicious, I think, thing that I get asked is, but can you really find people capable of doing this work in X or Y country? And this is around the time that I turn on my video at Zoom and say, do you really want to ask a person like me that question? Because you know the answer you're going to get. So before I dive into exactly how we've been working towards evening this playing field, I wanted to quickly run through the way in which we think about genomic diversity for conservation genomics, because there's a much broader audience listening. And so you can either think about it, at level 1, just genes, each thing coding for a simple trait. So this could be something like eye color, or in the context of species ID, a DNA barcoding gene. You can then look at collections of genes in multiple individuals to do population-level genetics, things like building pedigrees. Level 3 is the assembly of reference genomes. Now, these, at this stage, are usually incomplete. So maybe 1,000 or more scaffolds, but this is, as complete as we can get the single DNA code for one individual. And you can do all sorts of fun stuff, like comparing this and looking into the past in evolutionary genomics. But if you can really get to the level 4 side of things, where you can assemble things at the chromosomal level, then anything is possible. So I know I'm leaving RNA out, and there's a lot of fans of RNA here. I too think uracils are awesome, but in the interest of time, I'm just going to move through what we can do. So we have been looking at these 4 different ways of thinking about genetic and genomic diversity, and we've been trying to figure out ways to give people a chance to do this in their country of origin to solve problems that they think are important. And so this is where the In Situ Laboratory Initiative was created. It has 5 countries involved right now. People are using a variety of methods, so you could be thinking about this in terms of labs in a backpack, to labs in museums, or at field stations. And the range of problems they're trying to solve are enormous. So anything from SNP genotyping of chimpanzees in Rwanda to diet metabarcoding of multiple species in Peru. And I'm going to be able to talk today only about the results coming out of one of these hubs, because there's so much going on. And we figured we'd drill down into this. So this is the Wildlife Conservation Laboratory at Los Amigos. It's situated at the Estación Biológica Río Los Amigos, which is on the Madre Dios River. And it's an incredible site. There are over 4,000 species documented here. And if you look at the birds, there's 567 species on site. So anyone here that brought their binoculars to London, just in case you saw an exciting bird, which maybe I'm the only one, I don't know. This is the site for the world's big day record. So somebody that was able to see or hear over 350 bird species in a single 24-hour period at this site. In June of 2021, the WCL was launched after some tricky construction and renovation during the COVID pandemic. And by July 2022, we had collected about 7,000 specimens from a variety of sources, and I'll go through those in a second, per year. And we had done a lot of DNA barcoding with the help of what will now be sort of a team of 6 incredibly talented scientists. Some of our specimens come from rescue centers. So these are places that received injured animals, attempt to rehabilitate them. And this is an incredible way to look at the human-wildlife interface because of the variety of ways in which these animals end up, at these centers. But we also have an enormous mark recapture program. So this is a program that has been established at the site. It's been running since about 2018. And it looks at a vast range of taxa. Pretty much anything that will voluntarily enter a trap is captured, sampled, and released on the same day. So some great outputs. Reference library that's actually accurate, right? And that isn't something that you can take for granted in the Amazon. So we looked at everything from plants and herps to bats, primates, and other small mammals. And I'd really encourage you to please go and check out the poster. This is what it's titled. I don't know its number, I'm afraid. But for the results of this attempt, so including everything from multiplexing 1,200 samples on a single flow cell to having a sort of custom indexing, low-cost pipeline that the scientists in question would be really excited to share with you. We've also taken this up a notch. So one of the wonderful things about having traps that are open and permits to collect anything, a sample from anything that walks into them, is that you have an incredible biobank. So just in the span of those couple of years, we were able to get over 75 genera represented in this data set, and 178 fecal samples that we did diet and microbiome screening for. This is also in a poster. This is an online one, so please look for this title, and you can figure out how we were able to do this in an entirely solar-powered laboratory in the middle of the Amazon. So in August of 2022, something happened that sort of shook us. A very, very rare species walked into one of the traps. It was the end of the field season. We had given up on trapping, and so somebody forgot to clean the bait out of one of them. So fish went really, really bad. And apparently that's the short-eared dog's jam, right? So it's like, love this stuff, entered. By the end of that year, we had talked with Nanopore, and Org1 was just kicking off, and they were really excited to give us a P2 solo, which we flew in. There were several political disturbances in Peru that resulted in the P2 solo flying across the Andes multiple times, with libraries moving around from museums to the field station. But we were able to do some really exciting genomics. And since that day, we've worked with the genome of the short-eared dog here on the left, also a critically endangered primate, the yellow-tailed woolly monkey, of which there are only about 500 or so left, and they only found in Peru. And we've also been working with the Andean bear. So there's loads of these in captivity all over the United States and Europe. This is a bear cub from the San Diego Zoo, for example. But there's very little known about their population genomics, and we're doing some resequencing projects with those. So this is another poster that I would like you to all go and see. I'm just here repping an amazing team. So we were able to look at the short-eared dog and the yellow-tailed woolly monkey using the P2 Solo. And then we thought we'd throw in actually a bird with a smaller genome, only about 1 GB. And we ran this on just MinION flow cells. These are the results. They're already out of date, even though I assembled the slide at the last possible moment sometime last week. So we've got more data from Pipra, the bird. We're now up to about 30, I think, X coverage on it, and an 11 MB N50. So these are really great complete genomes, but they're all level 3. So for level 4, you need to do some Pore C, which is an adventure in the Amazon. So you can't get liquid nitrogen over the Andes, so you can't actually pulverize a tissue sample very well, although I tried. And so we've been working off of blood samples, and about a week and a half or so ago, we finally got the first Pore C data out, and we're attempting to base call and put that in as well. We were also lucky that about 3 days ago, a male short-eared dog walked into one of the traps, and so now we have a Y chromosome, which we're also going to be able to add to the mix. OK, so it's May of 2024, and we've had 3 reference genomes assembled at this tiny site. We've done 3 resequencing genomes for the Andean bear from Peruvian bears for the first time. This is the first ever Peruvian bears sequenced. And there are 5 plant reference genomes in the mix, which is the stuff of nightmares. But that's plants for you. And I wanted to just shout out to all of the incredible people that are able to make all of this possible. I know I mentioned Nobel Prizes before, and I seem to be sort of bringing them up rather often, but it's not because I really care to have one. But I think that if there were Rosalind Franklin's or Marie Curie's in the audience that had this in their future, that were ambitious enough, that wanted to achieve something truly stunning like that, maybe we should be thinking of them more in terms of not Franklin's and Curie's, but Caceres, right? You guys want to stand up? So Sanchez Vendizu or Sacco or Lipke, because they're the future and they're an incredible team of research scientists that I'm just really proud to be able to work with. And just in case I wasn't sort of clear enough, this stuff is infectious. So we set up one lab, and so many other people are excited about doing this in the same way. And I wanted to bring this out. This is from Rwanda.
Welcome to Rwanda Water Conservation Association Laboratory. Here is where magic happens. And here is PCR room. Samples are in here. This is clean room. Reagents are here. Thank you so much.
So I would just want to say not to let anyone tell you that you need a really fancy space. You can do labs and containers, which is what's going on there. And that's it. Thank you very much.